Okay, so as Brandon so nicely introduced me, I'm Todd Hayden. I work for the Great Lakes Fishery Commission. It's actually just changed not too long ago from Michigan State and the USGS. So technically, I'm working for the Great Lakes Fishery Commission these days. I live up in Sheboygan, Michigan. I'm stationed in Hammond Bay, and yes, we've had some floods here lately. I'm sure you've seen us in the news. So. Um, Super excited to be here tonight. And I'm really like to thank the Sea Grant for the opportunity to tell you a little bit about the acoustic telemetry program in the Great Lakes and end up here and talking a little bit about Saginaw Bay and some of the cool projects that are going on there and how we're using that information in the management realm. So that's sort of the where we're headed this evening. Again, if you need to or interested in more information or want to talk about telemetry or talk about fish, whatever, you can get me. Here's my email up on the screen. If you're interested more in the GLaDOS GLaDOS system, you can access the web page. Lots of great information there. And of course, you can always email the DNR. Or actually, the GLaDOS has its own web email too. So that's how you can get a hold of this if you have more questions down the road. So as Brandon sort of mentioned, the main questions and the main drivers behind the GLaDOS system is probably some questions that you may have pondered if you're an angler out in the water, right? The first one is why didn't I catch a fish today where I caught a fish yesterday? I've had this happen so many times. You go out there and you have a really great day, and you come back 2 days later, and suddenly the fish are gone. There's nothing happening. So it makes you wonder, well, these things really do know how to move or really capable of covering long distances, perhaps. And the other questions you may have experienced is you may have caught a tagged fish out in Saginaw Bay. For example, walleye jaw tagging has been going on for many years. And that's really one source of information that we use to manage the stock and manage the population. It's super important information, but it is limited in some ways. And that's sort of what I'm going to talk about today. Acoustic telemetry is sort of the next step past the sort of standard jaw tagging or Floyd tagging, whatever sort of tag you want to use to track fish movements and understand where they're at, where they're going. So lots of questions that we are interested in when we talk about fish tagging. Everything from where do the fish go? How long do they stay there? Where do they spawn? How long do they live? Do fish movements differ? Do males do something different than females? How deep are they? Do they cross state lines? Do they cross federal lines? Do they go back to the same place year after year? Do they know where they're going from one year to the next? And of course, what habitats do the fish prefer? And so these sorts of questions we can get at. These sorts of questions we can really get at a lot more detail using the acoustic telemetry. And so here's a few pictures. On the bottom left here, you'll see that's kind of a standard Floyd tag that's in the back of a walleye. This is sort of the old way of doing it. In the middle on the bottom, you'll see the different acoustic tags that we use to track fish. On the bottom right are receivers. So the idea between the acoustic telemetry is you put a tag in the fish, it beeps every few minutes with a coded signal that's then detected by the receivers. And those receivers log when that fish was there. And by virtue of knowing where that receiver's at, we can tell where that fish was. And then on the top right, there's a tag that's going into the belly, the abdomen of a walleye. So you can sort of see how this a lot of times how we tag fish for these studies. And so, as I just mentioned, the acoustic telemetry, you have this signal that radiates out from the tagged fish, and then it's detected on the acoustic receiver. And when it's detected, the receiver is a little computer. It logs the fish ID, the tag ID, and then also the time and date of when that occurred. And then the location of the receiver is assumed to be the position of the fish. Now, acoustic tags, you have different detection ranges based on the size of the tag and the strength of the tag. And so, you might get a detection range of 2 kilometers on the biggest tags, the strongest tags, anything, and then down to maybe 100 meters or so on the smallest tags, the least powerful ones. So that sort of gives you some sense of when we say, well, the fish is at a receiver, it's probably within a kilometer or 1,000 meters of that receiver at any given time. So one of the really nice things about acoustic telemetry versus traditional tagging is that you get multiple encounters per fish. So you can detect that fish. These batteries and these tags can last multiple years. So you can detect that fish over multiple seasons. And that's been probably the biggest eye-opening thing we've seen with these fish is that with acoustic telemetry is just how incredibly diverse the movements are and how incredibly repeated the movements often are. So it's just really interesting to be able to get that multiple year information. And this is, you just can't do this with your regular tags, right? If you affix a tag on the back of a fish and you put it in the water and then 3 months later, an angler catches it, well, that fish is likely out of the system and you don't know what it did in between when it was released and when it was detected. And then you don't know what it was going to do if it wasn't detected. And so the other thing that's really important is that this is fishery independent and that you don't have to have anglers out there fishing to be able to recover those tags. So we get information that's less biased by where fishermen like to fish, for example. And this provides a really good understanding or much more detailed information about movements of fish over time and space. So in this picture here, right in the middle of the screen, that's one walleye that we tagged down in Saginaw. Well, actually, no, this is one lake trout that was tagged up in Thunder Bay, a little bit north of Saginaw Bay. But you can see the red line sort of traces the movements over time. And you can see that fish was all over the lake. It was down in Saginaw Bay there off of Tawas and Augray, near Augray, and then all the way up in Les Chenaux. So this is the sort of information that we can expect out of these acoustic telemetry. Here is a walleye. Hopefully this is playing on the screen. I don't show the receivers in this, but you can see this fish is just all over the place. It was tagged in Saginaw Bay. It moves out of the Saginaw Bay, jumps around the main basin, goes back in Saginaw Bay, kind of makes a repeated sort of movement there. The color changes just showing time. So that shows you sort of again, the sort of information you can get from this system. So before I go any further, I wanted to talk a little bit about what the Great Lakes Acoustic Telemetry Observation System is. And basically what it is, it's a multinational network of researchers, and it includes state, provincial, federal, indigenous, academic, private organizations, anyone that wants to do acoustic telemetry, who are collaborating to use acoustic telemetry to understand the fish and address management needs. So currently the group is more than about 400 people. It includes about 107 active projects. Main funding is provided by the Great Lakes Fisheries Commission. And really the main purpose of the Great Lakes, the GLaDOS, the Great Lakes Acoustic Telemetry Observation System, is to share fish detections. So what often happens is that I tag a fish, for example, in Saginaw Bay, and that fish, and I put receivers in to track that fish, but by golly, that fish moves out of my receivers onto other people's receivers, receivers that are put in other places in the lakes that are for other projects. And so by having the GLaDOS system, that provides information back to me. We all contribute our detections for our tags, and then that information about what that fish did when it moved out of my acoustic receivers that I'm operating is returned to me. So this is it's like a value added sort of program in that you put in some receivers, I put in some tags, and you get information about fish movements across the entire basin. So it's sort of leveraging all these different projects and all these different people and researchers to be able to get much, much more information and to better understand the fish that we're tracking. So if you're interested, the GLaDOS webpage is a really interesting place to go to visit. You can find all the different projects, both the old projects that have finished up, as well as new projects that are coming online. And what I really like to look at when I'm on the website is just where the receivers are at. And in this figure here, in this picture here, you can see all those yellow dots are acoustic receivers stationed all over the Great Lakes. So you can see if you tag a walleye in Saginaw Bay, it's a you got a pretty good chance of discovering if it's going out of the Saginaw Bay into Lake Huron, or even moving into Lake Michigan, or even moving down the Detroit St. Clair system into Lake Erie. So it provides a basin-wide sort of view of what the fish may be doing. Um, Lake Superior, it's coming along. Lake Superior is a kind of a different beast because it's such a huge body of water. Hard to get all those receivers out there, but there are plans here in the near future to really boost up the number of acoustic receivers that are in that system. Lake Michigan, the same way. Here in the next year or so, if you look at this map in another year or so, you'll probably see a lot more acoustic receivers down towards the southern end of Lake Michigan. So now what I want to do is I want to take just a couple minutes to talk about a couple of the projects that are ongoing in Saginaw Bay. Walleye's one of them. Walleye project that we've been working on here, myself, along with a whole host of other people, Dave Fielder, Andrew Briggs, a lot of Michigan DNR folks. And the idea is we want to know where the fish are hanging out during the spawning period. So the thought has been for years that really the majority of walleye are spawning or hanging out in the tributaries around Saginaw Bay. Um, but what we want to do is to take and test that, right? So the idea is if we tag fish in the bay during the early spring and we track them year round, we would be able to take and determine what rivers they would go into or what reefs even, what area of Saginaw Bay they would be going to during the spawning period. We can't actually tell where they spawn. So that's one thing we haven't quite figured out in the technology is that there's no sensor that can tell us, yep, this fish spawned right here. But what we can do is just infer spawning perhaps by where they're at in the bay or in the rivers. So in 2022 and 2023 in May, we tagged 350 fish out in the bay. These were all mature walleye, 3 years and older, and we tracked them for 3 years. And so this is just winding down. We're in the sort of the final analyses, writing up some manuscripts and reports to publish from off of the results or the results from the study. All of the fish were tagged on recreational boats. So the DNR was able to team up with a whole bunch of recreational fishers down in Saginaw Bay and able to take and basically just use their boat for the day to tag fish. So they catch a fish, they tag it right on the boat, and they put it back into the lake, back in Saginaw Bay, right near where they caught it. And so on the bottom here, pictures, you've got a fishing boat out there in Saginaw Bay, and this in the bottom to the right there's a couple pictures of the actual surgeries that were happening on the boat while they were fishing. And then on the far left, there's a figure of Saginaw Bay, some red and blue dots. The red is 2022 and blue is 2023. That's where all the fish were caught and where they were tagged. So you can see we span the entire bay, caught a lot of fish all over the bay and tried to distribute those over space and time as best we could. And I think we were pretty successful with that. So, okay, the other part of this, where are the acoustic receivers in Saginaw Bay? And on the left figure here, there's a picture of an underwater picture of an acoustic receiver. They're suspended off the bottom. There's no buoys on the surface usually. That's just to keep them out of harm's way mostly. And what we oftentimes do is we use a grapple, a hook to basically snag a line that's lying on the bottom. If you're an angler and you're ever out there and you snag a rope and it doesn't seem to want to come off the bottom, please put it back. Don't cut the rope because that creates a lot of problem for us. It could be one of our receivers. In the middle figure here, you can see sort of the close-up of Saginaw Bay. We had receivers all over the bay. And then we also had receivers on the mouths of all the tributaries, especially the Tittabawassee, Saginaw River, the Gray River, all of the main tributaries that we have known spawning runs in. And then on the far right, we have figure of all the receivers that were in the bay during the project that we used to identify where those fish were during the spawning period, which we defined as March 15th to April 15th. All right, so the take home here, the interesting part of this whole study, before the study, the general thought, and this is based on lots of observations over many years, was that about 2% of the Saginaw Bay walleye, um, and 2% of the walleye in Saginaw Bay tend to be in the spawning season during the spawning season tend to be in Saginaw Bay, and 98% inhabit tributaries of Saginaw Bay. So that's the Tittabawassee, the Saginaw, the Flint, all those tributaries. But what we found in the study here, it's about 65% of the fish stayed in Saginaw Bay during that March 15th to April 15th period. So, and 35% were in the tributaries. So this has huge potential management implications. Maybe we need to think more about Saginaw Bay reef restoration. And maybe the tributaries aren't as important in the overall population as we thought. So this is kind of a huge result that's going to impact, I think, the management side of things for quite a while here. So that's sort of the latest and greatest on the walleye in Saginaw Bay. Now I want to talk about sort of a different study that we are also in Saginaw Bay, but it's a kind of a different objective. There's a movement of happening to take and reintroduce Cisco in Saginaw Bay. Cisco are really they're a native species. They're a really valuable food source for walleye and other predators in Saginaw Bay. And they're also really good to eat when they get to be big enough. So there's a recreational opportunity there too. So the recovery portion of this is all based on hatchery reared fish. The idea is you rear these fish in the hatchery, you stock them into Saginaw Bay. Hopefully some of them survive, and then we're able to take and rebuild the population. So our study here was really to understand whether or not these fish that we're stocking into Saginaw Bay are able to survive and do they move. So these fish are stocked when they're about 6-7 inches long. So they're just the right size for a hungry predator fish. And the question is whether or not these hatchery fish have any hope of surviving, really. And you might be asking, well, why would you want to do this? And it's a high energy prey resource, very good for the bay food web. And there's commercial and recreational fisheries certainly that could be very popular and very lucrative in the future. And since you might be asking too, well, why would you think about doing this now? Successful habitat restoration has happened in the last 50 years. So the habitat is a much better place and much more likely to be able to support these populations. Invasive species were one of the reasons that historically the Cisco disappeared from Saginaw Bay. And so the invasive species have been better controlled as well. Walleye in Saginaw Bay are hungry and need something to eat, so it would provide a nice food source for those as well. And as I mentioned, this is all based on using hatchery culture and then the future stocking of these fish, these Cisco. So we did sort of a proof of concept study, and the idea was to determine, can we tag and can we track these fish? Cisco in the hatchery are maybe 6 inches long. Small fish get to be very difficult to tag. It's just there, and if you've ever handled a Cisco or any of those sort of fish, they just aren't known to be really hardy. They're very, very sensitive to handling problems. So we were able to, in the hatchery, we were able to get tags in 25, 26 fish. They then were released in Saginaw Bay. Here on my bottom left, there's a figure of a map. The star marks the spot. They were released there near the Au Gres area. In the middle figure, we put in acoustic receivers around the release point. And they range from about 1 kilometer, that was the distance from the release, all the way out to about 4 kilometers. And so what we found is that 7 Cisco were immediately, within a couple days, consumed. So they were eaten. And how we know this is that we use these new tags that are able to detect when a predator eats them. What they have is they have a little composite sensor on them that dissolves in the stomach acid of predator fish, and it changes the signal. So it tells us when it goes by a receiver that, oh, it was actually the fish, the predator fish that swam by the receiver, and that the fish was consumed. So this is a really powerful bit of information to know that, yes, that fish was consumed, and we aren't actually tracking movements of a big walleye that ate the Cisco that we're trying to track. So that's super helpful. And then we had 13 fish that immigrated. And what I mean by that is they jetted out of the array and disappeared into the system within about 4 to 5 days of release. Half of them were gone within 4 to 5 days of the release. And then we had 6 fish that we really just can't tell where they went. So the take-home story here is that these hatchery-reared fish, they moved much quicker than we expected. They didn't hang out near the release location. We didn't know what to expect, really, whether they would move quickly or if they would stay pretty close to the release location. We also discovered, and this wasn't a big surprise, predation is an important consideration. These juvenile Cisco were definitely consumed. And we also learned that it's possible to tag and track these small fish in these 6 to 7 inches. That was probably about one of the very first studies that's tried to tag fish that small in the Great Lakes, certainly. And so this has really important management implications as well. When you start to think about stocking programs, the number of fish that you can need to stock to reach your goal, right? Reestablish the population. If you can keep a few more alive each time you stock by knowing something about what they're going to do when you release them, then that means you can stock fewer fish, and that means you can raise fewer fish overall, and that means you can decrease your cost. So there's lots of implications here for those sort of management decisions. What's really interesting is it allows us to take and start to explore, does it make more sense to stock these fish at night, or stock these fish during the day, or stock these fish in deep water versus shallow water? We can start to, with this new technology and the ability to tag and track these fish, it gives us an opportunity here to take and learn a lot about what works and what doesn't work when it comes to stocking. So other projects in Saginaw Bay. So I don't have time to go into the whole list, but there are definitely lots of other projects that are happening in Saginaw Bay right now. Jeff Jolly and April Morey both have juvenile lake sturgeon projects. There's a large lake whitefish movement that's heavily using the receivers in Saginaw Bay. And also, of course, there's a really cool brown trout project that's happening in Saginaw Bay. And there are also these many other projects that are in the planning stage. Hopefully, in the next few years, will be kicking off. If you want to learn more about the projects that I wasn't able to talk about, you can find information there on the GLaDOS webpage. So, with that, if I have time, I'm happy to take any questions. And go from there.
Wonderful. Thank you, Todd. We appreciate you virtually joining us to share that amazing information.